Okay, everyone, I think we'll go ahead and get started. If when you walked in, there were some handouts. If you grabbed them, great, but if you didn't, you can always grab them on the way out. First, I just want to welcome you to the session on genetic therapies and non-modulator-based therapies. I think listening to Kara's presentation earlier was a great primer. I think we're going to get into a little more details on some of those different approaches. Just to introduce myself, I'm John Sheridan. I'm a research director with the CF Foundation. I manage our academic research grant portfolio in our Path to a Cure space. And I'm joined by Patrick Thibodeau, Vice President of Basic Research, who will introduce himself when he comes up here to present. So we're going to do a little bit of a tag team presentation. So this is just a little bit of our roadmap of what we're going to cover today. So I think it's important that we first talk a little bit more of the basics of the lung anatomy. As Kara said, we're really focusing on the lung primarily as our first target. So maybe going a little bit deeper into understanding some of the complexity of the lung, I think is an important thing to review. As well as getting into a little more the biology of DNA and RNA and that protein synthesis process. We're going to cover a little bit of the modulator space and how they function. And then really getting into more of the non-responsive mutations and how they function, and covering genetic therapies, as well as the big barrier ultimately is the delivery side of things that we'll cover at the end as well. So this is a cartoon of basically what the lung tree looks like, all the way from the trachea at the very top, all the way down to the alveolar space where gas exchange occurs. And the cells that line your airway, the anatomy of that looks different as you move down to smaller and smaller and smaller airways, ultimately to where you reach the alveoli. So this is just a highlighted image of kind of a cross-section of what your epithelium looks like. So this is more your primary bronchi. So it's a very large airway. And what you see is that it's a very thick tissue. So at the top here, I don't know if I have, can see the mouse? Yeah. At the top you see is this layer of the airway epithelium. It's what we call a pseudostratified airway epithelium. There's ciliated cells, there's mucus up top that flows and helps allow for that mucociliary clearance process. I like this image because it also really highlights submucosal glands that are part of the larger airways in humans. You can actually see one here kind of moving off of the airway into the submucosal gland, and other components of this like cartilage in the airway that's really important, as well as blood vessels that feed the nutrients to the airway from the basal side of the cells. But if you move down a little bit smaller, and this is where we're getting into more of the bronchioles of the airway epithelium, you see that it looks a little bit different. We still have the airway epithelium. It doesn't quite look the same. It's not quite as thick. There's also other things like smooth muscle that help with contracting the airways, as well as you can see other evidence of blood vessels. But then you move really to that terminal end where we have a terminal bronchiole, and this is just a cross-section, so you can see how it's a very simple epithelium, and it ultimately leads into the alveolar space, which are all this kind of white open space. Importantly, you see a lot of capillaries and blood vessels around because that's where gas exchange occurs as well. So the main point of this I wanted to make is that the airway architecture is complex and it varies throughout the lung. So it's important that we keep that in mind as we're thinking about what cells do we target, how do we deliver to these cells, because it's not necessarily a one-size-fits-all approach. So if we now just take a quicker look, dive into the actual epithelium itself. This is quite a colorful cartoon here, but I think it does a good job at highlighting cellular diversity in your airway epithelium. Not all cells are the same. They all have different functions and different roles. So there's really kind of four main types of cells. There's some other extremely rare cells as well that aren't here. But there's the basal stem cells that Kara mentioned. Those give rise ultimately to all the other cells. There's secretory cells, which are in this orangish reddish color. as well as ciliated cells, which are important for that mucociliary clearance. And then the ionocyte, that's a really rare cell type, but expresses a large portion of CFTR in the airway. About 50% of the CFTR found in the airway is expressed in the ionocytes, and the other 50% is found in secretory cells. So this just shows how the basal cells, they are the stem cells, they differentiate based on different signaling cues that are found in the airway and turn into these different cell populations. So if we target those basal cells with a gene editing therapy, they should ultimately give rise to all the other cells that are found in the airway epithelium. Okay, so let's cover the DNA to mRNA to protein process a little bit more. So as we know, DNA is really our genetic code. It contains all of the genes that make us who we are and includes CFTR. So in the nucleus, it undergoes transcription. That makes the mRNA, and the mRNA leaves the nucleus, where it's then translated into protein. And so that's kind of this is the normal process that should occur. We go from DNA to mRNA to protein, in this case, the CFTR protein. So properly, it functions as an ion channel, allowing chloride to leave the cell. Another important concept that I think we really need to focus on a bit as we move forward in the presentation, and I think some of this was alluded to in Kara's as well, presentation, is around the concept of exons and introns and kind of what they are, because some of the therapeutic strategies target different aspects of the DNA. So CFTR itself is made up of 27 exons and 26 introns. So your DNA is basically interspaced with these introns. When that gets made into RNA, it looks exactly the same, essentially. But this RNA is then processed by the cells, and it removes all of the introns to only have you're left with the exons at the end of the day. And the exons are what make the protein. So they undergo translation, and you get your functional protein at the end of the day. So CFTR is, the gene itself is very large. We're talking 189,000 nucleotides. At the end, after splicing, you have 4,440 nucleotides. So a lot of the genetic information in there is ultimately cut out to result in your functional protein. So I put this up. It's there's a lot of information on here, and it's not important that you read through this, but it highlights that there are different types of mutations that result in different outcomes. And importantly, you can have mutations in the exons, which are a lot of the ones we know, like the F50ADEL, G551D, W1282X, or you can have others that are in the introns, like 3849 plus 10KB, C to T, that typically result in splicing mutations and other factors that disrupt the ability to properly process that mRNA into the end product. So if we take a look of how one of these mutations affects the protein ultimately. So we'll just start with F50ADEL. So you have your mutation in your DNA, it's retained in the mRNA, and ultimately it's retained in the protein. But in this case, because of that mutation, the protein can't properly fold into its structure that it needs to be in order to function properly. And as a result, the cell recognizes this and degrades the product. So it misfolds. If we look at G551D, it's similar in the sense that you have DNA to mRNA with the mutation, and ultimately you have a protein where it does fold correctly, it does transfer and move to the airway surface, but it's not able to open properly. So you end up with the same result, there's no chloride efflux. So that's where modulators came in. So modulators act upon the protein. That's really the important concept here. In the case, there's two flavors of modulators. There's correctors, and then there's potentiators. Correctors really help the protein ultimately fold into its correct conformation. So as your mRNA is being translated by the ribosome, the machinery that does that, it then folds properly by adding the corrector. And then adding in a potentiator helps that protein ultimately open properly. So Trikafta is made up of the three drugs that are different correctors and potentiators as well. So again, modulators act on protein. They don't impact the DNA, so they're not genetic therapies. So you've already seen this slide a bunch already. But essentially, in the US at least, we think roughly 94% of the population could ultimately benefit from highly effective modulators, leaving that 6 to 10% that don't have access to modulators and will need some other form of genetic-based therapy. So I'm going to turn it over to Patrick now, who's going to cover and run us through a lot of the different strategies that we're exploring to advance these therapeutics. Thanks. Patrick? Oops.
Thanks, John. Thanks for setting the stage. I think we had a great introduction with the earlier session, and John did a good job of explaining some of the basic biology. Just to let people know, I have been working on CF in basic research for almost 25 years. Finished the last of my academic responsibilities yesterday about noon, actually, so it's a bittersweet moment. But I think what I'd like everybody to do is take a pen and paper out. We're going to have a pop quiz on what John went over. Yeah? Okay. I didn't think anybody would bite on that one. They didn't in class either. So, I think one of the things is, as we heard this morning about the pivot to genetic therapies, is we're thinking about really different space to address the needs of people whose genetic background doesn't produce protein or produces protein that doesn't respond to the modulator therapies. And the pivot to that is to move into the genetic therapies, the nucleotide-based therapies. And really, there's a big list of things in here. There's RNA and DNA-focused therapies. And one of the biggest things that I think was highlighted in the session by both Steve and Kara this morning was the fact that RNA therapies are transient therapies. We can deliver the RNA. We know the RNA is capable of producing protein, but the RNA turns over in the cell. In contrast, DNA-based strategies tend to be less transient. They're a bit more permanent. We have DNA-based therapies that can exist potentially for months to years in the body, and then we can have permanent therapies at the level of nucleic acids based on DNA modulation that are truly permanent. And if you ever want to remember that, the names give you that indication. RNA is replaceable in the cell, and DNA is durable. Or DNA is also dinosaur. We used to talk about this in the lab, that we can dig up things that have been in the ground for millions of years, and we can get DNA out of it. DNA is very, very durable. It's very robust. So ultimately, I think as we think about moving through this genetic therapy space, the target is to try and do this at the DNA level. This is we've talked over years about sort of the one-time cure, and I think we'd all really like to see this, that you would go in, you'd have a single therapy or a single course of therapies that would persist for the person over their entire lifetime. That likely will require us to be in the DNA space and not in the RNA space. But as we also just heard, the RNA space is where we are right now. We have therapies that are coming into the clinic. We've all had RNA therapies, as Kara pointed out, with our COVID vaccines. And I think we're going to have data on this in the very short term that really help us bridge the gap while we develop some of these more advanced technologies in the DNA space. So it's this tremendously exciting path as we move forward in this space, where the RNA basically gives us our first step. We move through the DNA, and we can ascend Mount Everest, as Steve showed so elegantly in his slides just a few minutes ago. So let's talk just very briefly about a couple of the different kinds of mutations that are not modulator responsive and how we're going about addressing those. And of course, nonsense mutations we've heard a lot about. Nonsense mutations occur when we have a lesion in the DNA or a variation in the DNA. Whoops, I apologize. Let's see. Moving the mouse and advancing the slides, it's the same thing. But where we have a lesion in the DNA which introduces a premature stop. So we don't make the entire protein. This occurs in the DNA. When the DNA is transcribed, it becomes an RNA molecule. And when the RNA is translated, we basically get an incomplete protein. And this looks something like this if we just take sort of a cartoon schematized view of this, that we start with the DNA, the gene at the top. This is first transcribed, and we make what we call the primary transcript, where we have both the exon and the intronic sequences. That then goes through a process where we splice the protein, and you'll see the little red bar that's indicative, in this case, of the 1282X or the 1282 stop mutation, passes all the way through this, down into the messenger RNA. And the result is when we make the protein, we get an incomplete protein, and really at the same time, there's a very sensitive system in the cell that's aware of this kind of mutation, and we degrade the messenger RNA. So not only do we make a partial CFTR molecule, but the template, the RNA itself, is degraded. So we don't get much of this. So there's strategies to deal with this. John talked very briefly about antisense oligonucleotides. We've got programs that are entering the clinic with this, where in very select cases, what we can use is we can use a sequence that binds the messenger RNA and changes this process of splicing. So if we think again about the 1282X mutation, and we take the DNA, the chromosomal DNA, we make the RNA, we have the red lesion, in this case in exon 23. If we don't do anything to this process, we'll end up with that mutation carrying through in the messenger RNA and the protein, as I showed in the previous slide. With an antisense oligonucleotide, in some cases, we can actually block the splicing event at those sites. The result is, instead of making a transcript, a messenger RNA that contains exon 22, 23, and 24, we can skip 23 altogether. The result is we miss a little chunk of the protein, but in some very specific cases, we get functional protein at the end. And so we can actually remove that small piece of CFTR and develop enough protein with probably some other modulator therapies that we can have functional CFTR at the cell surface. And so, this is a very exciting approach for us to be thinking about. It's going to work on a very select number of mutations. This is a very specific thing that has to happen. And the analogy is we can think about this as sort of making a car, right? We think of cars as having four wheels and being quite stable. And in this case, if we remove one of the wheels, we can have a functional car, but you don't want to go around corners too quick because it's not particularly stable and it'll roll over. And that's sort of what happens to the protein, by the way. The protein is not fully restored to that of the full-length piece. We're still missing a wheel. It's functional and it may get you there, but you have to be cautious about how you drive with it. This is an exciting space, though, because this approach is being used in other genetic diseases. In the case of Duchenne's muscular dystrophy, there's an antisense oligonucleotide that induces exactly this kind of exon skipping and provides relief for patients that otherwise would suffer at a very young age of a very debilitating disease. And we're not going to go into that, but I think one of the really important things in this space is that we're taking these kinds of learnings from other disease indications and other research programs and really trying to apply those to CF so we can accelerate our own efforts in this space. So we also have splice mutations. And you're going to see a lot of the same sort of cartoons here, so hopefully it gets easier to follow. But this is much the same thing, where we have the DNA is at the very top line there. And in one of these intronic sequences that otherwise sort of goes away when we go through this process of splicing, we have a variation. The 3849 plus 10 kb is much like this, where we basically have a lesion that otherwise doesn't code for the protein, but we know it affects the production of the protein. So we can do exactly the same thing here, where we can basically, normally we would have the RNA before splicing, and what would happen in a situation like this is you'll see that instead of splicing out of that center exon into the final exon, so the blue box here and the one on the far right, we basically splice into this gray site and we pick up this sequence. This makes a protein that has a bunch of junk in the middle of it, basically. And proteins typically don't like that. You can't just randomly insert pieces in the middle of them. The result is a lot of these are non-functional. So we can take exactly the same approach when we think about the antisense oligonucleotide that we used for exon skipping, and we can mask the sequence that's the non-coding variant in the intron between exon B and C here. And so it looks much like I said before. We have a lesion that goes through from the DNA to the RNA, the primary transcript. We can then include an antisense oligonucleotide that masks that sequence, and the result then after the splicing process is that we can restore the normal splicing of the protein, basically restore the production of a fully functional protein. So again, these are specific treatments for very specific mutations and variations, but we're seeing in the clinic that these work in other indications. We have good preclinical programs in this space for CF. We're really excited about this as being able to deliver changes in these sorts of splice regulation events for our community as well. So I want to shift gears a little bit. You know, what we've done with modulators, and effectively we do, is very similar to what we do with the ASOs. And in fact, you could think about antisense oligonucleotides as being modulators that work on RNA, right? They don't change the RNA itself. It's the same piece of RNA, but we're doing something by binding a molecule to that piece of RNA that then changes its function. What we're going to shift gears, and we're going to think about actually replacing the coding region of CFTR over the next couple of slides. And this includes details on the messenger RNA replacement strategies that were highlighted earlier, as well as some very simplified schematics of how we think about going into the chromosome and replacing CFTR at the DNA level. So this really durable replacement of CFTR. So messenger RNA therapy, you know, in this case, we've got our pretty little epithelial cells that are in the airway. We have, these are, they don't look much like CF cells in this case because we've illustrated them as being hydrated on top with mucociliary clearance, the little black arrow, but this is the goal that we want to get to, right? So we basically develop a particle that encapsulates either, in this case, we're going to encapsulate messenger RNA that gets delivered into the cell, and then we can express the protein. That messenger RNA, as Kara had said, the advantage of doing that is we can make thousands and thousands of copies of CFTR off one messenger RNA molecule. So we only need to get a few of those into the right cells to make enough protein where we can get function. So this is really one of the chief advantages of using a nucleic acid-based therapy versus a protein-based therapy. And you can see that we think about this, the initial targets will be lung epithelial cells. Most of the programs are looking at aerosolized delivery to the airway. The delivery will be a CFTR messenger RNA molecule. We think the duration of this is probably days to perhaps a week. It's going to be short-lived because the messenger RNA, remember, is RNA, so it's replaceable in the cell and it will turn over. And the outcome of this will be short-term restoration of CFTR protein. For people with any variant, I think this is really the important thing, we're putting a new copy of CFTR in with this messenger RNA, and it doesn't matter what's then on the chromosome in the DNA, that we can give a good copy of CFTR into these cells. So we have a similar approach where we think about gene replacement therapy or DNA therapy, where instead of delivering a messenger RNA, we're going to deliver a piece of DNA. And it starts the same way with some delivery vehicle that encapsulates the nucleic acids. Those get delivered to the cells. They can either be delivered from the airway side, which is what was just shown on the cartoon there, or from the blood side. And I think this is something we'll be exploring more and more with the DNA approaches, probably more so than the RNA approaches. The chief advantage of this, because you'll see most of these sort of target, the target and the therapy other than being DNA and RNA, the big thing is the duration is much longer. We can transiently give DNA to cells and it will persist in some cases from weeks to months. That depends on the tissue, it depends on the type of DNA. But the advantage is you would have a therapy that instead of having to take daily or every few days, in the case of messenger RNA, that it might be monthly, bi-monthly, you know, at some extended period of time, and that's yet to be determined. We don't know what that will be, but this is the thinking about the differentiation, the principal differentiation between messenger RNA and DNA. So there is something, if we sort of hone in on this, we have the same delivery from the slide, and if we zoom in on these cells, what we can see is in the nucleus of the cells, down here, we have a copy of our chromosome, and the little red dot is supposed to indicate a broken CFTR molecule or a mutated CFTR. We take our delivery particles. This is actually a multi-step process when we talk about the DNA. The delivery particles have to go in the cells. The delivery particles then have to dissolve, and they liberate their DNA. And so the DNA is this green circle here. Through a process that is not totally well-defined for a lot of these specific events, that DNA has to make its way from not only inside the cell, but actually into the nucleus where our chromosomes are. And that's a very important step. And then at that point, that green DNA can start to produce its own messenger RNA. And it's basically we're now making 2 copies of CFTR in the cell. We still have the copy that comes off the chromosome, and that's sort of denoted as these purple bars at the top of the cell. Oops, excuse me. But at the same time, we're making a green copy of CFTR. And that's shown also sort of in the 2 cells where we had our delivery particles actually enter. That is the functional CFTR that comes from the DNA that we actually delivered. Due to the way CFTR works, this is okay. We're actually, it's not a problem to have a broken CFTR and a functional CFTR. We don't actually have to worry about the broken CFTR so much as just getting the functional CFTR in the cell. And we think this is part of the reason that this strategy will work. So then we shift gears a little bit to probably what will be that next step that Steve laid out earlier in our pipeline, our therapeutic pipeline. And that's actually going in and fixing the mutations in the chromosomal DNA, in the genes themselves. So not augmenting what's there, but actually fixing the primary mutation. And it's the same way we start with our delivery particles. In this case, these may be delivering nucleic acids. They also may be delivering proteins. And this is a very exciting space. The technology has evolved in the editing world over the last 10 years at an amazing pace. It's almost every day there's a new technology, there's a new strategy to make these tools better, more efficient, and more specific. And I think as we watch that space and engage with these companies and the academic researchers, we're seeing tremendous progress that we'll be able to apply to CF in the near term. The advantage of this is, if we target the right cells here, we can edit the chromosomal CFTR, and you'll notice now that the duration of this therapy went from some short period of time to basically the lifetime of an edited cell. And if we can target the basal cells, if we target the progenitor cells that gives rise to the tissue that is our lung, that basically means that's the lifetime of the individual, the patient. And so this is the idea of the one-time cure, that we would go in, fix the problem in the gene, and then allow that to basically continue through their life. Their cells would become normal, if you would, if we can efficiently revert the mutations in the chromosomal copy of CFTR. There we go. I like it. You get extra credit on that pop quiz that's coming. So, this is also in some cases a fairly mutation-specific event. We're going to go back to the model that we showed a few slides ago where we have this mutation, in this case W1282X, that's shown in the middle of exon 23. Basically, what we have to do for gene editing is develop a typewriter, a DNA typewriter, that goes in and literally rewrites the code, right? That changes those 4 nucleotides, the A, T, G, and C, back from the mutated state to the normal state. And so there's a tremendous amount of work that's been done developing these typewriters. Probably you've all heard about CRISPR-Cas over the few years. This is the one that's made the literature. There's a huge family of proteins that are now being engaged in this space. And each of these makes a slightly different typewriter, right? That allows us to go in and very specifically change these mutations in the gene and fix them. And it's almost like you're taking the white out in the gene when you've got your typo, you're erasing the mistake, and then you're fixing that typographic error in the DNA itself. So that again, the copy of the DNA is correct for us. So we develop that. These basically then go in, target the sites in the DNA, Now the DNA is fixed. You'll notice the mutation's just gone. When we make the messenger RNA out of this, we make a correct protein. And so that is really exciting work. The fact that that is going to be, I think coming online in a lot of disease indications is just really, really exciting as a researcher and as somebody thinking about genetic therapies. So the final strategy that we can think about employing is a gene insertion approach. And you've probably heard about this in other forums, but the idea here is that we basically can put a copy of CFTR into the CFTR locus and sort of ignore the pits that are not correct in our patient group. So we do the same thing. We've got some delivery particles. They go into the cell. This should be a hat. Again, these are, if this is done right with high efficiency, the duration of this is the lifetime of the edited cell. So it's another version of the of sort of the permanent one-time cure. One of the advantages of this is that this is not mutation specific. So if, much like the messenger RNA, if we can develop this and get this into the cell with high efficiency, it doesn't target individuals with a specific mutation, 1282X, 542X, Delta F. We can get everyone. We can bring everybody along with a therapy like this. And sort of very quickly, what does this look like? So here's our normal copy of CFTR on the chromosome. Each of these little red lines indicates the potential mutation that you'd find across the gene. What we do is we would engineer what we call a super exon, a coding piece of the DNA that in this case we're going to stick behind exon 1, which is the gray box shown on the far left. When we stick that in, and if we engineer this correctly, what we can do is we can get, struggling with the mouse here today, apologies. Yeah. What we can do is we can get the splicing event to occur between exon 1 and this super exon, which contains exons 2 through 27. The idea here now is that the protein that we would make is encoded by this small bit, which came from the original piece of DNA, and then this piece that we engineer that is the rest of the protein. And we effectively ignore all of the other things that are left on the backside of this. They're just never transcribed and translated into protein. So this is really exciting. We can approach a lot of mutations, and we can change the strategy and where we make the insertion site so that we have appropriate coverage for all with CF. So finally, I think just to touch briefly on therapeutic delivery, this is the biggest challenge we have right now. How do we get these things into cells? This is always a problem in drug development, getting the drugs to the right place at the right time, and it's no different for the genetic therapies. I think when we think about CF, there's a couple of strategies. First of all, we're obviously highly focused on the lung at this point, but we also know that CF is a multisystemic disease, and to provide that complete cure for CF, to fully resolve CF, we know we have to think about fixing this problem in multiple tissues. So there's two generalized approaches to delivery. The first is aerosolized delivery, where you would nebulize an agent, it would go into the lungs and then penetrate that tissue in the cells. And really the delivery control is this mechanism of it going straight into the lung. You're putting the drug in the lung by taking this approach, that's where it goes. The other way to think about this is a systemic delivery, where you would get something by IV and it would basically move all throughout your body. And in that case, the delivery mechanism, we have to engineer it to go to the right cells. We don't know that we actually want CFTR to go every cell in the body. We want it to go to the cells that need to make CFTR. And so there's a challenge with systemic delivery that we're taking on with many of these companies, where we're actually engineering what we call tropism. And tropism is just a fancy science word for meaning where it goes, if you will, where we could basically engineer the molecules, these particles, to go to very specific cells in very specific tissues and express CFTR where we want it to be, and not express it where we think it would be a liability for our physiology. And I'm not going to spend a lot of time on this. The challenge is that the airway, as you know, as both Kara pointed out and Mike pointed out, the airway, our lungs, are actually this amazing organ that has evolved to keep everything out. And we breathe in millions and millions of pathogens and particulates every day. And for most people, we clear those out. Even in the CF lung, that's the case, that there's some clearance. We also have lots of barriers, including immune cells, a mucus barrier, plugs associated with mucus, bacterial colonization, biofilms, et cetera. So there's a big barrier there. One of the really exciting things is working with academic labs and industry companies to develop these new technologies, new chemistries, new polymers to get across these barriers and ensure that we can get our therapeutics into these airway cells. And we do that. There's basically two groups of these. I think this was mentioned earlier. There's lots of ways to define this. One are viral vectors, and of course we've heard about viral vectors for gene therapy for a very, very long time. What's really changed in this space over time is the discovery of new vectors and now the ability to engineer these vectors so we can actually build on the outside of these viruses the zip codes that get those viruses to go to the right cells, right? So before we were relying on the virus's ability to just find the cells on their own, and sometimes that was good and sometimes that was bad. It depended on where you wanted the virus to go. And in this case, the technology's developed such that we can basically address those viruses to start going into the right tissues and the right cells. So the technology changes here are really amazing. At the same time, the delivery of messenger RNA and DNA components via nanoparticles, either lipid nanoparticles or other polymers, has increased. And that the chemical space, sort of the complexity of the things that we can look at, has expanded greatly over the last 10 or 15 years. We now know that we can control some of that chemistry. We know that there are rules that govern where these particle goes based on size and charge and some other properties that, again, we're able to build in, that we're basically able to engineer in that zip code to get these things to go to the right tissues. So it's really, really exciting space. And just finally, looking forward at the pipeline, we have a tremendous number of therapies in this space that are basically advancing every day. I think this slide is actually a little out of date compared to what was shown just a few minutes ago. That's actually how fast it's changing around here. And it's hard to keep up. These are molecules, many of which are in early stage clinical trials and include some of the genetic therapies. And of course, some of those have just opened and begun enrolling, some are ongoing, and we're expecting more over the next year to 18 months. And I think there's a lot to be looking forward to in this space. We have this extensive preclinical development program engaging companies with these really exciting technologies. And the breadth of what we're evaluating on a regular basis is really mind-blowing as a scientist. And then finally, as a plug to the part of the research that John and I do, is we're engaging really across the world the best academic researchers we can find. So in my mind, as a plug for academic research, I think when you get into clinical programs, you have to have a proof of principle that something's going to work. And Steve made this discussion or analogy to Star Trek. The academic researchers are often the ones that are thinking 10 years ahead. Here's a crazy idea, what if, right? And I think the role of the academic researcher and the people that we're engaging is to redefine the possibilities that we have for the development of these therapies. And I think in doing so, we are really, truly engaging people that, they're 3 steps ahead. They're thinking, what can we do if? And part of the support that we're able to bring is not only in the preclinical space that we heard a lot about, but to the labs where people have an idea that it's not a product yet, it's not a data point, it may be a crazy idea that's founded in an observation or in science. And I think those are the kinds of things that'll continue to expand the space that we have to operate in and bring us additional possibilities moving forward. I think I'll just stop here and we can take questions. Again, I think this is a very robust space. It's a very exciting time to be in this field and working at this intersection between academic and basic science and the preclinical development. I think working together closely with Steve and Kara and other members of the research affairs team, we're thinking about how we can be most innovative, how we can really accelerate these processes to bring these therapies to bear for our patients. And I guess the final comment I'd like to say is that that's the team that I see every day that I work with, but actually it's our team, right? Everything that we're able to do at the foundation is based on the hard work and the dedication that you as volunteers, as our families, as our advocates, as those that have communicated on our behalf for decades now. We are standing on the shoulders of you who are the giants in the field in many ways. And I think this is it's a wonderful community to be a part of. The opportunity to come work with the foundation after 20 years in my own academic lab working on CF was one I could not turn down. And I am just so excited and so honored to be a part of this community and be on your team and have you on our team as we move forward. Thank you.
Okay. Oh, Steve, go ahead. Great. Thanks, Steve. So you can so I didn't say at the beginning, and I apologize, I missed that, but you can submit questions through the app. But I'm happy to, if you have a question and want to shout it out.
I just want to second what Steve said. I am really excited. You guys discussed the hell out of me. I have a doctorate in physics. I never studied biology, but I'm getting more acquainted with it, and you're blowing my mind. So thank you. I really.
Great, thank you. So we do have one question in here, so let me, oh.
Yeah. We have a short list of sayings that I think we're picking up over. Beam me up, Scotty might have to join our list with the 11th commandment and some of Bob Bell's sayings that get passed around. I like that.
Yeah, so the one question we have here is, can you go into a bit more detail on the work SpliSense is doing? Do you want to speak to that?
Yeah, so the SpliSense program, again, to go back to academic programs, this started in an academic lab at a university where the observation was made that the antisense oligonucleotides could induce this sort of exon skipping, and at the same time that the protein that was made had some residual function that we could act on, and so it really was a path forward at really the most basic level. SpliSense has been one that we've been involved with for, it predates my time. I don't know when we initiated that as a preclinical program. Mike and Steve aren't giving me guidance back there either. Thanks, guys. But right.
Yeah.
But that's a program that we're actively engaged with and supporting right now. I think they have multiple indications and multiple ASOs that they've developed over the years to target CFTR. The preclinical data that we're seeing, I think, has been validated in our own lab as well as other labs. And so I would just say that that's a program we're actively supporting, and we're eager to continue to see the progress that they're making with the ASOs.
Great. I see you have a question.
Yeah, first of all, thank you so much for your but I'm going to ask the tough question. When I get home, how was the conference and when? So might be a question for those in the back room. Yeah.
No,
Well,
I mean, yeah. I mean, I think we have, therapies like a 4DMT is approaching. They're in the process of enrolling patients, and we're getting to the point where we're going to have some results and data from some of these other strategies, like their gene replacement approach, as well as other companies that are mRNA focused, like Arcturus Therapies, which is approaching the clinic, as well as another company called Recode that's also moving forward. I think the gene editing side of things is further down the road. It's challenging to do that approach of how to get all of the appropriate machinery that you need there to the right location. That's a significant challenge. And I think, as Steve mentioned with his kind of mountain analogy, there's a lot of these intermediary steps that are going to get us to that point. So I think it's going to be, I think every few years we're going to have advancements, as we move forward. But
Yeah, I guess. Yeah, well.
Thanks. Yeah, I mean, it's very hard to put dates on it because there are unknown unknowns. So you're asking about best case scenario. Well, we've got one enrolling, first patient in already. That's happening. We're trying to get about 20 patients in that study to make conclusions. How fast will it roll? How fast will they come? Will they have problems on the way? Assuming none of that happens, then I think 6 months to get that done, and then start the readout. If it's like a game-changing result, then we're looking at a study that will need to be about 60 patients in. Okay, so and 6 months between those to design that study, get it approved, get ready, et cetera. A 60-patient study, you could imagine it being global, hard endpoints, 3 to 6 month period, depending on how long we need and how those regulatory conversations go. So that's probably couple year effort. Then 6 months to get it approved, written up, make sure it's tight, everything works perfect. I mean, that's the best case, but a lot of risks along that way.
so why don't we go to you, and then we'll do another question on the tablet. Oh, okay.
I love that everyone in the foundation that we're willing to embrace crazy ideas, because that's how you advance science, and I'm very grateful for that approach, and I think we've seen the field from it. So here's a crazy idea. I know that it seems like you guys are saying that the lung itself poses so many problems with trying to get in these techniques, right? So my kid has CF liver disease is her primary thing. Has there been any thought about trying to pilot this on a smaller population where it might be easier to get in, like in the liver or in sinuses or GI? GI is a huge issue for most of our patients. And anyway, I just wanted to float that crazy idea. Thank you for listening.
Yeah, I think we have these conversations on a regular basis. We've engaged. There have been a number of companies that have come to us that have technologies that would target extrapulmonary tissues. Liver would be one that would probably be, if we went down that path, and I'm gonna put if we went for just a second there, but if we went down the path, liver is a tissue that's fairly easy to deliver to. I think there's some real complications about what do we need to do to affect the course of disease there. I think that's one of the big challenges as we've shifted in the clinical and basic research. I think we've been so focused on the airway for such a long time, and now we see that we can solve some of these problems for most of our patients, but it is a multisystemic disease. So what we really need to do in some of these spaces that you've just laid out is really engage new clinical scientists, new physician scientists that can help us understand what your family members are experiencing. We need to engage the right basic scientists so that we can then go and think about how we fix that problem and that we're doing the right thing. And then I think the technologies are there that we can implement in some of these cases. I know Kara and I have had many conversations about advocating for looking at therapies in the GI space, because the cells are, it's much easier to take things up in that space. The other is the cells turn over, so you can do things to those cells. If the cells don't really like it, maybe you'd be uncomfortable for a few days, but basically the cells would be replaced. It's really a question of balancing where we are and what the needs are, number one. And then I think also being really careful about knowing what the physiological changes that we need to go after. And I think right now I feel like that space, it's balanced between what we're doing preclinically and then huge investments in the academic, the basic research to really make sure we understand what it is that's causing CFLD, CFRD, the GI disorders, some of the bone health, the whole gamut of things. And that space that we haven't had as many researchers in over the years because we've been so focused on the airway. It was a huge priority for us.
Because I know in my daughter's case, her CF, the failure of her liver was caused by just chronic GI problems that would last for 20-something years. So there's so much we don't understand about the gut. And because of so much pulmonary focus, we may be just kind of missing such a big opportunity.
Well, we appreciate that. We're working on that very hard. That's been a huge investment that we've been making the last few years. We've also been doing this, I should say, with the NIH. And the NIH has made very significant commitments to studying CF outside the airway, thinking about those kinds of extrapulmonary problems that our community members have, because we need to address those, and we know we need to do that.
Great. So I have one more question here, and then we'll take another from the audience. So the question is, what's the relative difference between difficulty of delivery of mRNA versus DNA? And I think inherently, it comes down to what the package that it's being delivered in looks like. The DNA or mRNA inside that package doesn't necessarily matter. What matters is once it's in the cell, can that DNA get to the nucleus where it needs to function versus an mRNA that does not. So it's really designing the vehicle that's going to allow us to get there to have the proper ability to target the right cells and get taken into those cells. And that's where a lot of the challenge and that's where a lot of effort is being focused. Another question? Oh, you have the mic right now. Yep.
With over 2,000 variants of the CFTR, how do you make determinations on what variants you're going to be targeting for like mRNA or for DNA restructuring or replacement?
So again, for the messenger RNA delivery, we can almost ignore what the variant is. You're putting a new copy in, a new perfectly good copy of CFTR in. And I think, that the ability to work in a mutation agnostic way and be able to ignore what's there really powers some of that. I think some of the other discussions when we think about mutation-specific therapeutic development, we're really thinking about what's the unmet need that we have with the modulators. Where can we be the most impactful? Meaning what patient group or with those variants? Is it one person or is it 1,000? And that's part of that. There's another component of that is what is technologically possible right now. And as we engineer all these systems, particularly the ones that are going to be variant specific, there's some limitations that we don't know how to get around that are technical limitations, just the way these enzymes and the systems work. And that constrains a little bit of what we can do. And so the way we address that is that we work to try and engineer that out with all of the researchers. But I think there's a pretty complex calculus that goes into that. But I think, trying to be as impactful as possible with the effort that we put in for those that don't have access to therapies now, it drives the first part. And then I think the second part for us in some of that evaluation is what can we do with what we know we have in hand right now versus what is the next step. Again, I think this analogy that Steve laid out of going up this mountain, I think, that is really a multi-layer thing. And I think, we can target the quote unquote easy thing from a technology point of view, show that that's a proof of principle, then do the engineering and start working on some of the other things that are a little bit more difficult. So there was,
I don't know who. I guess,
What?
Kara, you had a question right there. Let's just go there.
Maybe you've touched on this already and I missed it, but when you talk about delivery of mRNA and gene delivery, I'm curious how what that looks like. Is it inhaled? Is it an oral pill? Is it a shot, an injection? How is this getting into the body?
Yeah, so I think a lot of our approach right now, especially in the industry side of what's advancing, are all these inhaled-based approaches. So it's going to be you're nebulized as a viral particle, essentially, that has the virus, which is just our delivery vehicle that has the appropriate cargo, or these nanoparticles, lipid nanoparticles, or these polymer-based approaches. And they have to then get through the mucus and the aspects. The systemic-based approaches for a lot of these are going to be the similar vehicles. They're going to be viral vectors or non-viral vectors, nanoparticles, that will be directly fed into the vasculature, the circulation of the body. And they will, ideally, if they're designed appropriately, home into the appropriate locations based on everything that's built into that vehicle, that will help it find that right location. Okay. Sure. Yeah. Sure. Yeah. Sure. Mhm. Sure. Great. That's a question.
I have a question that's more related to I guess, the business of what we're doing. And going back to, we're also grateful of Dr. Bell's vision at the time to change and go to a venture philanthropy model. As he always said, we need more shots on goal to really make progress. And I just commend, staring at this slide, the number of shots on goal that we have is just tremendous. And he must be really heartened by that and looking at it. But I guess my question relates to, I would assume the partnerships that we have with pharmaceutical companies are somewhat proprietary. And I was struck by when you put the slide up about the Duchenne syndrome and potentially learning from the research that was there. My question relates to how much sharing goes on when, I guess I'd call them breakthroughs occur relative to this genetic technology that's occurring, and how much of that is shared with the perspective of, if more is shared, progress can happen faster, because there's obviously researchers working in the same space that are not related to our particular community. So I'm just really curious how much sharing goes on and how restrictive is that based on the partnerships we have?
Well, I would say there's a couple of points there. I think the sharing, that these are corporate entities and they're, protecting their intellectual property rights. So some of them do take a more altruistic approach and are more generous with their sharing, and some are maybe a little bit less so. I think one thing that we can do in that space is, as we work with them and we partner with our own lab and our academics, we internalize a lot of that knowledge. And I think we're learning as these programs develop as well. I think the other part of that is engaging the academics. So a lot of this space, it's coming out of academic labs. And I think we have an opportunity to work with them where the technology is first, conceived before it's even commercialized. And in that case, I think we're getting insights into technologies and processes that we normally wouldn't have in a fully baked commercial company. I don't know if Steve or Mike want to comment on once we sort of get deeper into the development process, but I think it is an important point that if we can learn from our drug development, from one drug development company, and that we can facilitate in the next step with the next generation, it's really important. And I guess I would say we have an example of that where our lab did that, where we have the learning, which Steve pointed out was Translate Bio, which was a messenger RNA program that failed a couple of years ago. And we know now, looking back, we don't always have this, the foresight is never as good as the hindsight, right? We now know that, there are indications that we're looking for in our preclinical studies, in our lab studies, that allow us to gate whether we move forward or how we move forward or how we push on the technology based on the success or the failure that we've had in the past. And I think we learned from that program. We've pushed our programs to demonstrate, like was shown on the slide this morning, the production of messenger RNA in the relevant cells. And I think this is a case of sort of this evolution of learning. But I don't know if Mike wants to comment on specifically the corporate aspects of that.
Yeah, thank you. I might just add one thing. So, great question. Part of our job as well is truthfully staying aware of not only what's going on in CF, but throughout genetic technologies. So we've had several instances where we see some technology that looks really interesting that's being applied to other diseases, where we actually will ask to meet with those companies and to talk to them, to try to understand it more. And really the question then is this something we think could be applied to CF? Because if it is, that's a great signal for us to start to talk to them and say, we think this is something that could potentially work in lungs, or we would like to test this. Would you be willing to test this in our lab? Because we'd like to understand what this, because that actually allows us then to signal or to take that next step of saying, hey, we would like you to have a program in CF as well. So some of these companies, Recode's a perfect example. They're not just a CF company. They actually go broadly. They're trying to deliver across many different diseases. We're rooting for them to be successful in some of these other diseases and to have breakthroughs, because absolutely we'll take that then and say how much of that applies to CF. So it's an ongoing relationship. We have a whole team. I mean, you've seen these guys, but there's a whole team. There's a big part of what Kara does and others who are always thinking about, okay, what's going on in the science over here? Could this apply to CF over here? It's actually going to conferences that don't focus on CF a lot of times, but trying to pull that technology back. And then we have the best story to tell. We say, hey, look, this is why you want to do CF. We're going to help pay for it. We've got experts to help. We got a lab that can do it. When you want to do clinical trials, we've got a clinical trial network. That's where the story really, that ecosystem comes in. I don't know, Steve, is that anything you want to add there?
I think that was a great answer. All that being said, like, there's definitely a highly competitive space in genetic therapies. You can read the newspaper about even the competition on who gets the patent and go into a major court system, et cetera. So we have to negotiate that too, but we're savvy enough that we can start to broker these relationships to break those barriers down where we can to then engender partnerships, because they understand, we can help them, and they've got to share, and then we can help them, and they benefit too.
Great. Well, I think we're at time, but I'd be happy afterwards if anyone has additional questions to discuss as well. And we'll have another session this afternoon as well if you wanted to come back and see it all over again. But I really want to thank you all for coming in and for your questions and participation. We appreciate it. And Kara, thanks for being our mic runner.